This is an overview slide. It is not in your notes. I believe it's in the skeletal muscle notes, which are coming up. You don't need it. I'm just going to sort of like tell you where I'm at here. So we've talked about the resting potential. We've talked about the action potential. We've talked about how it propagates. It's been all review. Your information comes down to the spinal cord. It's transmitted to the muscle via action potentials. And your action potential hits this nerve terminal, and it causes the release of neurotransmitter into this neuromuscular junction, which is a specialized type of synapse, which we're going to talk about in some detail in this lesson. That depolarizes the membrane and causes an action potential to fire. It propagates through the sarcolemma down into the T-tubules as well. And as it propagates, it depolarizes the membrane. That causes calcium release into the cytosol. That binds to troponin, and that causes your skeletal muscle to contract. Right? That's the process that we're going through in these lessons, and we have reviewed this. And now we're going to concentrate on this, the synapses. Okay? And then in the next lesson, which will come with the next case, we'll talk about muscle, which will be some review because it's very similar to cardiac muscle, not entirely. All right, so there are two types of synapses. I'm going to start with the electrical synapses. You guys are already a little bit familiar with this, and I'm not going to concentrate upon them, but I do want to introduce the topic in that they are synapses. Electrical synapses are bidirectional. They're basically gap junctions. Okay, so you guys are familiar with these from cardiac muscle already. They're definitely, very common, always pretty much there in cardiac muscle. They sometimes show up in smooth muscle and some other tissue types as well. These are made up of hemichannels, which are made up of connexins. These are located in the membrane of two adjacent cells. The hemichannels come together and form one big channel that form a communication within the cell. These gap junctions are large in diameter. They're very good at electrical communication. Ions freely flow through here, and if you have an action potential propagating down this membrane, it will transfer to this cell, and the action potential will continue to propagate because of these electrical connections between the cells. Okay. However, more than just ions fit through these huge channels. Nutrients, metabolites, water, things like that also find their way through. So remember, we talked about cardiac muscle as being a functional syncytium. It's a syncytium meaning a whole bunch of cells that kind of act like one cell because of these connections, which allow all these things to go through the channels between cells. All right. Again, they are bidirectional. There is no synaptic delay. I will talk about that in a moment. Chemical synapses have a synaptic delay. And they are, again, most common in muscle, especially cardiac muscle, and I think you guys know that now. Okay? So chemical synapses are really pretty much what we're going to spend the rest of our time on here. They are unidirectional. So an electrical synapse is bidirectional in that an action potential can come this way and go to this cell on this side, or it can come down this cell and transfer to the cell on this side, either way. So, and the communication is bidirectional in terms of the metabolites in the water as well. It can go across those channels from one cell to the other in either direction. Chemical synapses are different. They transmit information in only one direction. So you have a presynaptic cell. In the case of our neuromuscular junction, that's the neuron. The presynaptic cell releases a neurotransmitter into a synaptic cleft, and that neurotransmitter binds to receptors on a postsynaptic cell. Okay? So you have an action potential in the presynaptic terminal that causes neurotransmitter release. That is released into the synaptic cleft. And then that binds to receptors on the postsynaptic membrane. In the case of a neuromuscular junction, that is the skeletal muscle membrane. Okay? And if it's a directly coupled synapse, your receptor is also your channel. So the ligand binds to the receptor, which is also the channel opens, all right? So these are ligand-gated channels, okay? And we call those directly coupled synapses when that happens. So a neurotransmitter binds basically to the channel. The channel is the receptor. The channel opens, ions flow into the cell, and that changes the voltage, okay? That's how directly coupled synapses work. Indirectly coupled synapses use second messenger systems, especially G protein coupled receptors are involved here. In this case, we have a neurotransmitter that binds a receptor that is not the channel, and then that will activate G proteins, okay? So in the case of the autonomic nervous system, we talked about activation of GS, which will stimulate adenylyl cyclase, which increases cyclic AMP, increases pKa. That's what we're talking about here. We're talking about G proteins that will either directly activate the channel or which will initiate a second messenger cascade, which will result in the activation of the channel. And the channel opens and ions flow into the channel, and you get ions flow through the channel, and you get a change in the membrane voltage in response to that. So these are indirectly coupled. Directly coupled, the receptor is the channel. Bind, it opens. It's that simple. Indirectly coupled use G proteins and second messengers, okay? So here's how neurotransmitter release works typically in a stereotypical synapse. So generally speaking, the neurotransmitter is synthesized in the nerve terminal, and it is stored in synaptic vesicles. So these are just membrane-bound vesicles in the terminal, and the neurotransmitter is waiting there in the vesicles to be released. You have an action potential that propagates down your neuron to your nerve terminal, and it depolarizes the membrane, which opens voltage-gated calcium channels. So we're familiar with these from the vital fluids and gases block. You guys know that calcium channels have activation gates. They flip open and they allow calcium to come through in a very similar way to the way sodium channels work. They're a lot slower than sodium channels, but nevertheless, it's very similar. These are N-type calcium channels. That's different from the T-type calcium channels and the L-type calcium channels that we have talked about in the past, but for our purposes, they operate the same. Activation, inactivation gates, the whole deal. So that channel opens, calcium flows through the channel into the nerve terminal, the calcium concentration goes up in the cytosol, and that causes synaptic vesicles to fuse with the presynaptic membrane, okay? And what we end up with is a process of exocytosis, where the vesicle fuses with the membrane, it dumps the neurotransmitter into the synaptic cleft. The cleft itself in a typical synapse is very, very narrow, about 200 angstroms, which is pretty small, as you guys know. And it can be more than that in the autonomic nervous system. The autonomic nervous system has a different purpose. It's releasing norepinephrine and acetylcholine into this synapse, but the neurotransmitter is just diffusing out along the membrane, and it's binding to receptors all over the place on the cell. This is different. This is a lot more local, and so a typical synapse, not in the autonomic nervous system, is quite a bit narrower, and so this release is quite local onto the postsynaptic membrane, okay? There is a synaptic delay, so I mentioned electrical synapses don't have this, of about half a millisecond to 1 millisecond. That seems to be mostly caused by the time it takes for the depolarization of the membrane to open these calcium channels. There's a little bit of delay there, and a significant amount of this is caused by that. So we have this very narrow gap, and because it's narrow, the neurotransmitter diffuses across it pretty quickly. and it binds to receptor molecules on the postsynaptic membrane, and that causes opening, usually opening of postsynaptic channels, and that will result in either excitation of this membrane or something that will inhibit excitation of this membrane. If it's an excitatory synapse, the voltage will go up and tend to try to fire an action potential or contribute to firing an action potential. If it's inhibitory, then it will resist that change. And we'll talk about both of those in this lesson, okay? But nevertheless, it is binding of the neurotransmitter and its action on these channels in the postsynaptic membrane that cause that, okay? So we have excitatory or inhibitory synapses. So let's talk about the neuromuscular junction. This is usually the first one that everyone learns. I won't say that it's prototypical, but it is a really common one, and it's a very good one to start with, okay? And in a movement and mechanics block, I'd say it's critical to know about it. So the first thing you should know is that these are extremely large, much larger than synapses of other types, like neuron-to-neuron synapses and stuff like that. 1,000 square microns is huge. This is an electron micrograph of one here. And of course, this is a idealized drawing here. The postsynaptic membrane, the skeletal muscle, is called the end plate. It has a special name, and it is quite large, okay? And there are a few dozen to about 2,000 skeletal muscle fibers per neuron, and the neurons usually creep along and synapse with individual muscle cells, one synapse per fiber. A few dozen, if you're talking about something that requires very fine motion and very fine control, maybe you don't want a whole lot of muscle fibers to be activated by one neuron. About 2,000, if you're talking about some big, gross strength maneuver where you just want to contract as many fibers as you can all at once, then this will be more likely in those kinds of situations. And that's why this variability is there. Okay, so we have one synapse per fiber, but we have a lot of fibers per neuron quite frequently. The neuromuscular junction, acetylcholine, is the neurotransmitter, and it is released from the presynaptic membrane onto the postsynaptic membrane here, the end plate. Okay. These usually have about 10,000 molecules per vesicle. One vesicle is called a quantum, okay? And one quantum causes what's called a miniature end plate potential. Occasionally, one of these will be released, you know, in the absence of any real stimulus, just, you know, occasionally, just spontaneously it'll happen. It'll cause a little bit of a depolarization here. The reason why that's significant for us is because there are several hundred of these that fuse simultaneously when you stimulate this fiber. Several hundred of these, each of which can cause a depolarization of 1 millivolt, means a huge depolarization. So there is a big depolarization in this end plate that is designed to really 100% cause an action potential to fire. You should hit you should hit threshold every single time. When Brian Urlacher is standing in front of me and he read something on my blog and he's really mad, I'm going to want to run, and I'm not going to want my neurons to try to stimulate my skeletal muscle, and my skeletal muscle go, maybe I'll contract, maybe I won't. No, it better contract, and it better contract every single time, okay? So there's no maybe about it. And these are designed so that it is all or none. There should be no maybe in a healthy neuromuscular junction. It should fire an action potential every single time. This should cause a huge depolarization, more than enough, with a big safety factor to cause an action potential in a skeletal muscle fiber. Okay? So the postsynaptic membrane receptors are acetylcholine receptors. As you might guess, these are nicotinic. They are different from the muscarinic receptors that we talked about in the autonomic nervous system, let's say on the SA node. where you're slowing heart rate down. These are different. They are called nicotinic. They bind nicotine. But they bind ACH, most importantly. They are located at the top of these junctional folds, very close to the presynaptic membrane. ACH diffuses across. They bind to these receptors, which are at very high density because, again, the system is designed to throw an action potential every single time. Okay, so that they're at the top of the junctional folds. There's acetylcholinesterase at the bottom of this fold, which is designed to terminate the response. So if you stop releasing acetylcholine, there's acetylcholinesterase here, which should suck up the ACH and terminate the response. Okay, and that's the reason why it's there. And it prevents something called desensitization. If ACH sticks around long enough here, the receptors will actually desensitize and they'll stop actually causing depolarization. They'll stop acting as receptors, period. And that is usually not a good thing, so you want to be sure and remove your acetylcholine in a timely fashion so that it's not just hanging around and binding to the receptor and staying bound all the time so that desensitization takes place. It is a ligand-gated channel, so this is a directly coupled synapse. The potential in a neuromuscular junction on the postsynaptic membrane has a special name. It is called an end plate potential. End plate, the postsynaptic membrane, in a skeletal muscle cell, potential because the voltage is going up. Okay, and note how big this is. This is what you get in a typical synapse. EPSP stands for excitatory postsynaptic potential. That's what you get in a regular nerve-nerve synapse, let's say in the central nervous system. This is what you get in a neuromuscular junction, a huge depolarization, because again, you have a lot of the ACH receptors. The area of the end plate is extremely large, which makes a lot of the high concentration here an even bigger factor because it's spread over a really large membrane. And again, it is designed to result in an action potential every single time. EPSPs, the excitatory postsynaptic membranes in the CNS, are not designed to do that. They are very small increases that have to summate to get to threshold before you actually fire an action potential. We'll talk about that in a minute, okay? So we have the end plate potential. It results in opening of sodium channels. That generates an action potential in the muscle. This depolarizes the membrane, and outside the end plate you have a bunch of sodium channels, and because you've depolarized the membrane, you hit threshold, these open, they cause an action potential, and you have the action potential that spreads over the surface of the membrane pretty rapidly. It's actually slower in T-tubules. There are actually fewer sodium channels, but what's really interesting is that a lot of this is caused because the T-tubule diameter is really small. So we talked about things that affect the conduction of action potentials, and the diameter of the fiber was one of those things. The bigger the diameter, the faster the action potential propagation. Well, in this case, it's not the cell that has a small diameter, it's the extracellular space, the T-tubule, that does, and that actually slows it down. So I think that's kind of interesting. It's like flipping the switch. We usually look at the diameter of the cell, but if you change the diameter of the extracellular space, you can do that too. Normally not relevant, but in a skeletal muscle fiber with T-tubules, it is. All right, I'm going to transition into the CNS system and the synapses in that system in the next video, and I will see you then.